In this chapter, we finally get to have some fun with DNA because we get to take a look at a very visual way of modeling a DNA strand as a worm-like chain model. So we are going to go through what a random walk model is, what a worm-like chain model is, and there are a lot of components that I have to draw out. So there will be a component where I am just discussing the different concepts, and there are different components where I'll be drawing on my tablet so that you can take a look at how it all comes together. So in the previous chapter, we took a look at different ways of modeling DNA. This chapter will focus specifically on the worm-like chain model. And with the worm-like chain model, we can then take a look at DNA elasticity, bending, and denaturation, and some other things. So once we discuss the worm-like chain model, we'll be able to apply it to DNA as well and discuss some of the mathematics that go behind looking at the energetics and the bending modulus of DNA. So we can model DNA in many different ways, and we are going to be looking at the majority of this. But like I said before in the previous chapter, we're going to focus on the charged rod, and we're going to focus on the worm-like chain model through an elasticity. So this chapter is a little bit long because we are going to focus on the elastic rod model and the random walk worm-like chain model. So for the worm-like chain model for DNA, there are many different things we have to go through. First, we have to figure out how DNA can be modeled as a worm-like chain. Then we can take a look at the random walk model chain of DNA. Once we have that random walk model sorted out, we can then model each individual component of our DNA persistent length as an entropic spring to take a look at its bending modulus and its elasticity. So once we have that, then we can take a look at different properties of DNA as the length extends and see how it can possibly wrap around a histone and how it can bend in different shapes and forms. So we're going to start off with the worm-like chain model. First of all, we have to understand what it is. The worm-like chain, or WLC model, is basically used in polymer physics quite frequently. It helps us describe the behavior of certain partially flexible polymeric chains. So what this semi-flexible or partially flexible definition refers to is the fact that we have a persistent length or persistence length. It means that your polymer is going to be fairly stiff if you have different segments pointing in the same direction. If you have a bunch of different segments that are pointing in different directions, then you end up with a little bit more flexibility. So here I have two pictures, one on the left of a linear rod and one on the right of a bent rod. So we're going to use these two to describe just different versions of a worm-like chain model. So I'll just label this as the linear rod. And this as the bent rod. They are going to have different qualities, and it means that we're going to have to label them in certain ways. First of all, we know that the rod that we're looking at is going to have a certain length. So here, we're simply going to say that we have a length L that goes across like this, from center to center. How can we model this though? One thing that we can do is if we do have a rod that's blue like this, we could potentially redraw this and create a central axis. I'm just going to draw one up here. So we could have a central, this being the central axis. Of what we're trying to model, which is DNA. So we'll call this central axis of DNA. We are going to assume that there is a DNA double helix, which makes sense. So on the top part, we have one strand, and on the bottom part, we have another strand. And because there is some sort of elasticity to these helix, double helix structures, we can model them as entropic springs as well. So we can do something like this. Okay, so there's a DNA double helix. Each one of these represents one of the helical strands. So we have K being equal to a spring constant of DNA strand of single DNA strand. There also has to be some sort of width to this, so we can consider this to be Z. And we also have to consider the length of each of these springs, and we can call this, let's put this in purple, A naught. So that's one way of modeling just a segment of DNA. We can consider a linear rod worm-like chain model where you basically have no bending taking place. Everything is aligned properly with all the persistence legs chained together. Now, what happens with the bent model? Basically, now we have to consider our axis L, or our central axis, to be like this. Just give me 1 second to... So that central axis has a length of L. So it hasn't changed because it's the same length, it's just bent. So in this model, we have to be a little bit more careful about how we model this. I'm going to extend this into a pie shape. So I'm just going to extend, I'm going to draw a vertical line here. That. Like this, just so that I can have some sort of angle here that I label and I call, oops, theta for the angle of bending, let's say. Now we know that L is here, so when we are modeling something, we can consider that as a length. And if I just extend it a little bit, like this, I can consider this to be my central axis of DNA. Just like before, now the axis is bent, and we're just simply going to call this L. So I don't want to make this diagram more busy than it is. I've essentially expanded this so that I have an example up top. It means that I now have my upper DNA single strand and my lower DNA single strand. They're going to be a little bit extended. So here I have something like this, approximately. So I have two of these, and it is going to be a change in length. So the length of the DNA strand up top is going to be a little bit longer than the DNA strand at the bottom. So we are going to consider the top length to be called L plus some sort of delta L. And we're going to consider this bottom length to be L minus delta L. So the assumption is that whatever change in length takes place up top, which is the amount that it has been stretched, it's going to be the amount that has been compressed for the bottom strand. So the other thing to note is this is some sort of radius that we can refer to as well. And that's about it. This basically helps us represent two different ways of representing DNA as entropic springs. So these are two ways of presenting DNA as entropic springs. Now that we have this model, we're capable of figuring out a way to measure and find the change in length, being able to figure out the bending modulus, and so on and so forth. So the next few slides will focus on deriving equations for just that. The first thing we want to find is delta L. How much bending occurs will depend on how much the top component is stretched and how compressed the bottom part is. So delta L is going to give us the amount that the length changes because of the bending effect. And so looking at this, we know that we can take a look at theta and have similar triangles in effect here. So I just want to carefully relabel this. I want to call this R. Make this a little better. Just to clarify what that radius is, it goes straight to the central axis of DNA. And we know from the previous diagram in green, we have the width as Z. So I'm going to label this too. So it means that when we have a diagram like this, the angle created for theta is going to be equal to L plus delta L divided by R plus Z, being equal to just L over R. So I'm just going to carry that over to the next slide. Change in length is considered delta L. Using similar triangles, we have L plus delta L divided by R plus Z is equal to L over R. And this is based off of similar triangles. Sharing angle theta. So based on that, we can solve for delta L. We have L plus delta L times R is equal to L R plus Z. We can expand and end up with this. See if we can cancel some components out. We know that LR will basically just cancel each other out. And then we end up with delta L being equal to ZL divided by R. We can highlight this and see that we've now defined delta L The next step is to look at the spring constant. And just to simplify matters, we would call this the DNA spring constant, K. So when we're trying to look for the total spring constant, we can use all the knowledge that we've covered in previous courses in first year physics. So to find the spring constant, we know that we have to find some sort of K total for the total spring constant of some sort of polymer chain or DNA. So K total we'll define as the spring constant of one strand of DNA, and that will just be like this. Something we've drawn before. We'll call each individual spring individual. So that means we'll use the subscript individual, and that means that each of these individual springs will have a spring constant of K, I, and D for K individual. We also know that there is some sort of length, A naught, that we discussed before. So K total is then going to be our individual spring constant divided by the number of springs. And that gives us K divided by the total length, especially if this is linear, the total length divided by how long each length is to get the total number of springs that are present. So overall, we have K over L over A naught, and this means that K total is going to be equal to K times A naught divided by L. So now we have two equations. We know we have the spring constant. We know we have delta L. With these two, it's now highly possible to be able to calculate the bending energy of DNA. So the next slide has to do with finding the bending energy, which we call E bend. So let's say we define E bend as the bending energy So from previous courses, you probably know how to figure out the potential energy of a spring. That is exactly how we're going to calculate E bend. We know that there is going to be two springs, so there's going to be twice as much energy as one. So 2 times, and this one you should be familiar with, 1/2 kx squared. But in this case, we're going to use K total, and x is the length, but we have delta L as the change in length, and that's going to be squared. So I'll simply ask here, where does this come from? This comes from Hooke's law for springs. We know that force is equal to negative kx. And we also know that the energy of a spring is going to be 1/2 kx squared, where this is K total in our case, and this is delta L in our case. So rewriting everything, we end up with E bend as 2 times 1/2 is simply just 1. So we would actually have, let me just say, I do want to keep this 1/2 out. So I am taking the 1/2 out just so that I can rewrite this in a certain way afterwards. We're going to put the 2 inside K total. We're going to put delta L squared separately. From the previous slides, we can redefine K total as Ka naught over L. And we can redefine delta L as ZL over R squared. Now, if we rearrange some terms, then we're going to end up with E bend being equal to 1/2 2 Ka naught Z squared. You can try to figure this out yourself. I have the Z squared over here. I'm moving it over to this side. And then on this one, I have L over R squared. So I've simply expanded everything and rearranged everything to look like this. Now, why did I write this in this particular way? So let me just... This portion is actually called Kappa B. Kappa B represents a microscopic entity. So these are parameters for the DNA worm-like chain model that help define its microscopic properties. Now, on the flip side, we have this one, L over R squared, which we'll put in purple. And this simply just represents the macroscopic entity. So these are terms that we can find out on a macroscopic level, like the length and the radius, that we can easily measure. But with the K and the A naught and the Z squared, those are smaller quantities that are much harder to measure. So just as an aside, our kappa B has another name. Kappa B is also called the bending modulus. And is defined by microscopic quantities. Okay, so now we want to move on to an analysis of the worm-like chain model. We were able to figure out the bending energy for one particular component of a chain, but what happens if we look at the entire chain as a whole? So first of all, I'm going to rewrite the bending energy as 1/2 kappa B instead of the entire collection of microscopic parameters. We're just going to use kappa B and then L over R squared. From 2 slides ago, when we first introduced the bending or the bent rod model, we also noted that theta was approximately equal to L over R. So we know theta is approximately equal to L over R by some approximation. Because of this, we can say E bend is equal to kappa B over 2, and then we have theta squared over L. Now, if you want to rewrite this in terms of R, you can also say this is kappa B over 2, and then you have theta over R, like this. So either version works depending on whether you have the quantity L or the quantity R for your macroscopic entity. Now, this particular bending energy only works for one particular R value and one particular length value. And when you're looking at an entire worm-like chain model like this red squiggly line over here, it doesn't work for the whole thing. So that means you have to split up the worm-like chain into different components. So this only works for one bend. And if we were to calculate the total bending energy, which we will call just E total, we have E total being equal to the summation of 1/2 of kappa B of Li over Ri squared. So we basically have to add up all of the different bending energies for each bending component of your worm-like chain. Another way to write this for integrals would be kappa B over 2 times the integral from 0 to the total length of 1 over R squared S ds for a certain distance away from the start. So let me just highlight this. Okay, so that gives us our total bending energy. Now we want to be able to figure out how to model this graphically. So I'll just name this the total E-bend for a worm-like chain model. So over here, the I's represent one bend. By one bend, I mean something like this. So if I were to take a look at the curvature here, I see there is some sort of curve taking place along this line. And so I know that there is a particular length from here to here that I'm just going to cut off. And I'm going to model it with a circle. So I'll use black for this. And so here we have the radius of A. Let's say this is the first distance A that we're looking at. So that would be the length of A from here all the way to here. Now the next part of the bend is going to be a little bit more, a little bit bigger. So let's use green. Could probably model this as something like this, just up until this point, because I'm assuming this loop can be modeled as one entire circle. So just do something like this. Oops, that's way off. So something like this, and it ends off here. So now we have some sort of length LB. Like that. And we have a radius of B. So now I've taken into consideration two bends within the worm-like chain model. The next one is going to be here. So I'm just going to assume this is like a perfect circle. Like that. And so the length would go to here, and we would have an entire loop based off of this. So this would be RC. And you would have the length being along this, being LC. Somewhere like that. And the final one is simply going to be here. From this region up until here. And so this is going to be length D for the fourth component. And there you now have 4 different regions, A, B, C, and D, for your worm-like chain model that can be modeled with different radii of curvature, and thus can be used to calculate the total bending energy of a worm-like chain. So just keep that in mind. This is a very visual way of being able to calculate those energies. And moving forward, we'll take a closer look at persistence lengths. So moving on to persistent length, now that we know how to model a worm-like chain based on the amount of bending that takes place, we now have to determine how much bending will take place. So that has to do with a discussion of persistent length. And so that persistent length, LP, is the length scale within which the chain remains straight. So basically, a good way of remembering this is how long can your chain be without bending? And that is going to define your LP. Beyond that threshold of LP, your chain is going to coil up and you start getting that worm-like chain structure. So there are 3 options to determine whether your chain will coil up or not, and that has to do with the change in length with your upper and lower DNA, single-stranded DNA in the double helix. So based off of our original discussions, we had a linear rod. So let's take a look here. This is considered a linear rod model. And if we were to model this as a DNA double helix, remember we have our central axis, which has a distance of L. We'll call it that. And we have a single strand of DNA at the bottom and a single strand of DNA at the top to form that double helix. So on the top and bottom, one thing to remember is the length here for the top part is going to be L plus delta L, and the bottom part is going to be L minus delta L. Now, if delta L is less than LP, then we can consider this delta L to be very close to 0, and therefore it's negligible. So if your change in length is much smaller than the persistent length, then you don't have enough bending taking place or enough change taking place between the bottom and the top single-stranded DNA strands to cause some sort of bend. As a result, you end up with a linear rod system. What happens though if you have delta L being approximately equal to LP? So at that point, let's draw this out. I'm going to start off with this. The central axis, always a good place to start with. So that is L. If I have delta L being approximately equal to LP, it means that I have to have some sort of extended component up here. And then a compressed component here. Like that. And this is going to form our shape. Oops. It's like this. Like that. So at this point, we have, oops, I need the red. We have L plus delta L, or basically this is equal to L plus LP. And then here we have L minus delta L, which is equal to L minus LP. What this means is we've just breached the threshold. We know that the length is small anyways, and so if you are extending the top and compressing the bottom by a persistent length value, it means that your little segment over here has just started to bend. So this is called the just bending model. Which marks the transition between a linear system and a fully bent coil-like system. So in a perfectly circular case, we'd have delta L being equal to LP, which is approximately equal to the radius of curvature. Now for the third option, what happens if your delta L is much greater than the persistent length? So in this case, there's only one option, and that's the option we presented before with the worm-like chain coil. So something like this, because you have a chain that's bending so much that it can no longer sustain a linear shape. So this is called a coil like WLC model. So this is basically a model where it is no longer circular or linear, like the previous two options. So these three different definitions of persistent length, as well as delta L for the change in length for each single-stranded DNA, that is very important to remember moving forward because it'll help us determine what kind of bending takes place and to what degree. Okay, so the next part we're going to deal with has to do with finding the persistent length. That's usually the next question. If we know what values will cause what kind of bending to take place, we need to know how to find those values themselves. So now that we know there are three options for types of bending due to a given value of persistent length and delta L, we do want to be able to define what LP is, or the persistent length. Without knowing the persistent length, we don't know if our delta L is going to be less than, greater than, or equal to that value. So how do we find persistent length? First of all, we do want to figure out the energy of bending for that perfect model of just bending in the middle. So focus on the just bending model. Where R is approximately equal to LP, which is also approximately equal to delta L. So in this case, what we do is we take a look at the bending energy for the LP case. And now I just want to rename that as E of persistent length, or we can just label this as ELP. And that's going to be 1/2 kappa B. We have Li over Ri squared. We know that Li is going to be LP. So we just write down kappa B over 2. We know this is LP, and we know also that R for this case is going to be LP. So we can simplify this by making everything the persistent length. Therefore, ELP is equal to kappa B over 2 LP. Now, the other thing to note is that this particular value can also be equated to something else. I can equate, or we can equate ELP to some sort of entropic value, which we'll say is KBT. So that is going to be because the probability of this particular shape taking place is proportional to e to the power of negative e bend, usually some sort of energy penalty, divided by KBT, or the thermodynamic beta. So once you have this, we know that there is some sort of relationship between the two of them, and therefore we have E bend of this shape being approximately equal to KBT. So based on this, we can simply say that kappa B over 2 LP is equal to KBT. And therefore, we can solve for the persistent length. So the persistent length is then going to be equal to kappa B over KBT. So this is actually the mathematical expression for the persistent length. So I'll just highlight this because it's important. So we'll just label this as the persistent length of a worm-like chain model. Okay, so next, we've basically gone through all different components of being able to calculate, we've gone through all different methods of calculating different components of the worm-like chain model and its parameters. Now I want to move on to different cases we should consider. And so in case number A, we have what happens when the length is less than LP, in which case you have a rod-like linear DNA structure. What you end up seeing is something along this line. In option B, you have the length being approximately equal to LP, in which case you have both effects being comparable. So you usually end up with a curved rod. And so that curved rod is simply just the same thing as before, but it looks like this. And we've seen this many times already. So something like that. And of course, the last option is going to be when the length is greater than LP, in which case you have an entropic coil taking place because it's no longer energetically favorable for it to maintain a perfectly bent structure. So in this case, you end up with just a coil system. Like that. So as a summary of the worm-like chain model, something we want to note is that if we are taking a look at a coil, we're just going to draw a circle here because it's easier. I have a starting point here. So we'll call this the start. And maybe we have something like this, and the worm-like chain bends exactly along this curve. And we can have this end like this. Okay, so the total length of this entire chain from start to end is going to be L. And we are going to consider a short distance S over here, or a point S at this point. And this will be defined as RS. So once we have this, we know that the energy of bending is equal to kappa B over 2 times the integral from 0 to L of 1 over R2, with R being defined at 1 point along our length. And we derive with respect to that those points. Next, we have LP being equal to kappa B over KBT. So these are the two most important equations to remember moving forward. Some key points to remember would be as follows. Basically, we can predict bending configurations by comparing delta L to LP. We can also group and define microscopic versus macroscopic parameters. And finally, we are able to find the total bending energy, or E-bend, for long chains. So that's a high-level summary of the worm-like chain model. You will be expected to know how to graphically represent this and do a few calculations based off of these equations. Now we're going to move on to the random walk chain model to see how we can model DNA in a slightly different manner. So moving on to the random walk chain model, we want to redefine this by looking at what it means. A random walk model just means that when you're taking a look at a lattice structure, you have a start point and an end point, and the direction taken for each step along the lattice is random. So let's say that we start over here. So we'll call this the starting point. And let's say we have to end up over here. Call this the end. It means that if I were to define a way for me to get from the start to the finish, I start here and there is a 1 in 4 chance that I can go up, down, left, or right. So let's go right, like this. Once I get to this point, I have a 25% chance of going up, down, left, or right as well. So let's just slowly work our way up. So a random walk model would be something like this, where each time it's going to be a random choice. Just draw a few of them, just like this. Up, left, up, right, down, so on and so forth. So looking at this, it's really clear that it's taking a well-wound path along the lattice to get to the end point. But the overall distance from start to finish, if we're only looking at the x-axis, and let's assume that the x-axis is only along this line, then we know for sure that the total distance is just going to be from here to here. But in order to get from start to finish along the x-axis from, let's say, point A to point B, we have to have n steps. And those steps can be in any direction based off of how many steps it takes to get there. So that's one way of taking a look at a random walk chain model. Another way to represent this is through just one line. So this would be the lattice method, and this would be the distance method. Different textbooks call it different things, but this is the one that sticks with me the most because one has to do with a grid-like structure and the other one has to do with just one horizontal line. So for this horizontal line, we basically have a start and a finish, or an end. And we know that the distance between these two will be x, just like in the previous example. But the way to represent this is a little bit different. Let's say that we have 9 steps. Then essentially I would start here. And let's say I go 1, 2, 3, and then somehow I go from here 4, 5, 6, 7, 8. So 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, so what this tells me is I have a start and an end, and overall the total distance that I've traveled is from here to here, and we call this a total traveled distance of xi. So these are just different ways of representing a random walk chain model that you can use moving forward. It's just a very graphical way of representing this. Now, a good way of being able to quantify different models using random walk would be to look at multiplicity. So the next step is to take a look at probability. So we're going to look at multiplicity W of nn, where we have n being equal to the number of steps to the right. And just remember, we are focusing on two-state systems. So basically, when these steps take place, the steps can only be forward or backwards or left and right. So n minus n will then be the number of steps to the left. And then capital N would be the total number of steps taken. So what would the multiplicity be? We have Wn being equal to n factorial over little n factorial, and then our difference between the two factorial. But there is something else. We know that x is going to be equal to, especially if n is equal to, if we know that one step is equal to 1 unit of distance, then we know that the total distance traveled would be n number of steps towards the right minus n steps towards the left. This will give us x equals to 2n minus capital N. And then we can figure out n to be x plus capital N over 2. And we have n minus n being equal to n minus x over 2. Just so we have different ways of representing these values. So using all this, we can say that Wn is going to be equal to n factorial divided by a quantity that can tie us back to the length that, or the distance that has been traveled. We have little n being equal to x plus n over 2 factorial. And we have capital N minus n being equal to n minus x over 2 factorial. So this is just another way of representing the multiplicity of the random walk model. So I'll highlight this version. This is the important one to remember. Now, the next question is, what happens if you have many, many steps? So our question is, what happens when n is a lot greater than 1? So what's the purpose of all this talk about multiplicity and everything? The purpose is to be able to figure out another parameter that we can use to represent the DNA that we're looking at. And that is the spring constant of DNA. When we talk about multiplicity, we always talk about entropy and energy as well. So the first thing we want to do is take that knowledge of multiplicity, the fact that if n is greater than 1, as in if we take more than one step, what happens? We can model it as a constant, use this constant here that's represented, and then also make another assumption in that with every step that we take, there's a step length of b. So then your total length is going to be n times b. So based off of this, we can update our equation for multiplicity to be this constant, to be e to the power of negative 1/2 x squared over nb squared. Once again, I'm not deriving this. And that can also be equivalent to e to the power of the negative free energy over kBt, where the free energy is equal to negative kBt ln of w. So based off of this, we can try to figure out the spring constant of DNA. And the trick really is to compare the energy that we have from multiplicity to the energy of a spring. So let's try doing this. We have multiplicity. Entropy being represented as 1/2 x squared over nb squared times kBt to give us some sort of energy term. And we're going to compare it to a spring. So this would be a mechanical spring. Where we have 1/2 kx squared, where k is equal to a mechanical spring constant. Now, obviously, if we're taking a look at DNA, we can't have a mechanical spring taking place. We have to have almost like a theoretical spring based off of entropy, and therefore a theoretical spring constant. So if I try to rearrange this, I'm going to get 1/2, and then I can have kBt over nb squared, and then I have x squared. So the goal is to make it look like this. So I'm going to isolate k, and so I can isolate this, and I still have some form that has 1/2 kx squared. So this over here in the middle is called, so this is called the effective spring constant of DNA. And so we define that to be K EFF is equal to kBt over nb squared. Now, this particular spring constant is important to remember moving forward. It is a spring constant that's based off of multiplicity and entropy. So in effect, it is some sort of entropic spring constant that represents how stiff a DNA chain is. So we can move forward. What happens if we take a look at an entropic spring with an external force? So I'm going to draw something here. Let's say that we have an entropic spring or some sort of chain of DNA, just looks like this. And it's being pulled down by some sort of force F. So if we have an entropic spring, and we call it entropic because it's all modeled based off of multiplicity and energy for the DNA worm-like chain model. Then what happens? We know that we must balance the restoring force, which is essentially negative kf times x with an external force F. So this basically means we are going to apply Hooke's law to an entropic spring. So we end up with F being equal to kfx. And we're just taking the magnitude here. So we have x being equal to F over k EFF. And we can also conclude that x is proportional. So the amount of stretching is proportional to the amount of force being applied. So this helps us to relate the displacement with the applied force. just going to highlight this over here. Okay. The next thing that we're going to talk about is chain elasticity. So now that we have all of these different parameters, it's time to put things together and be able to work on a few models that can be applied to DNA. So here I want to present one model that has been used by researchers since 1995. Back then, J. Marco and E. Sigia discovered this particular model for DNA, the worm-like chain model, as well as chain elasticity. So what this means is we can use this particular equation to model different components of DNA. I'll draw a diagram first, and then we'll be able to discuss what each aspect is. So we first have some sort of chain like this. We're going to consider L to be the length of the chain. So that's simply just L. We then have, X being the distance, horizontal distance from start to finish, or linear displacement of DNA start end. Essentially, that can be the distance between the 5' and the 3' ends. We then have F being the external force applied. And then we have LP being the persistent length. So based off of this model and this particular diagram, this is a worm-like chain model that is used to calculate different components of the system that you're looking at. So if you were to try and figure out the persistent length and you were to perform an experiment and you know the values of T, X, L, as well as the force applied, then you can theoretically calculate the persistent length and do further analysis based off of that persistent length to see which regions are going to bend in certain ways when you take a look at delta L. Okay, so next, I want to discuss what happens when you have a small external force F. So if we have a small F value, then it means based off of that model, you end up with 1 minus x over L to the power of -2 is approximately equal to 1 minus 2 x over L. That's then going to be approximately equal to 1 plus 2x over L if x is a lot smaller than 1. So this then leads to the following. We have x over L being approximately equal to 2LP over 3 kBt of F, which we know from before, and we know that the effective spring constant is equal to kBt over nb squared. When you combine the two of these together, you end up with K EFF is equal to 3 kBt over 2 LPL. So that is one way of representing this. And if we have B being equal to 2 times LP, being the step size, and L being equal to N times B, then we have KEFF being equal to 3 KBT over LB. So these are two different ways of simplifying that model to be able to calculate the effective spring constant for a polymer chain, or in this case for DNA. So we've taken a look at what happens if we can assume a really small external force, and we can also see how we can simplify the parameters so that we can redefine the effective spring constant. Now it's time to take a look at what happens if we increase the external force to a much larger value. So for a large F, we must have X getting closer and closer to L. So that means it's going to be fully stretched. So based on that entropic spring we were discussing before, if you have a coil-like structure and the worm-like chain model for DNA, if you apply a really strong external force, you're going to stretch the spring until it reaches its full length L. And so we say for our large F, X, which is the linear displacement, will approach the total length L. So in this case, how do we model it? For a large L, I'm just going to draw another diagram here. We have this being the start, and we're simply going to have something like this. And we have the end over here. Now, for the persistent length, I am going to split this up into tiny pieces. So something like this. This is our persistent length, the smallest length that can be considered a linear rod. And this is just approximate. So we know that over here, I'm just going to say this is LP. We do have to note, or remember, that B, which is one step size, is equal to 2LP. So that means we have from the start to finish, this being B, so on and so forth. We'll just finish that up. Like that. So we consider this B as a step size because it is approximately 2LP. Anything past this refers to a random direction of movement. So if we were to take a look and graph the trend of a worm-like train model, we'll have something like this, where we have X over L, the ratio of displacement over the total length, and the force applied. If you have a small displacement over the total length, then you're using a small amount of force. The more force you have, then the greater the displacement over the total length. So in this slide, I basically want to go through a balanced scale. A graph is presented here of chain entropy. The vertical axis refers to the probability of something taking place at a certain length, and the horizontal axis refers to that length on a ratio of total length divided by the persistent length. So what this is trying to tell us is that if we take a look at the peak here, like this, there's going to be a point where there's a very high probability, and this value is actually approximately 0.08, still very small. And this value is approximately 3.4. This peak is going to tell us that our energy and entropy are well balanced. So this means at exactly a length of approximately 3.4, there is a very high probability of maximum entropy taking place. So this is an optimal partnership between probability and length. So the trend basically states that higher probability of DNA step sizes or lengths will occur when energy and entropy are balanced. So essentially, the take-home message from here is that if you do have a particular length that is preferred and at a high probability of occurrence, then you know that at that point, the entropy is going to be at a maximum, and that both the energy available in the system and the entropy there are going to be well balanced and optimized to occur with high probability.